Welcome to the Expert Series, and thank you for joining us today. We're going to get started in just a minute. In the meantime, I do want to make sure everyone has their microphones muted, so just double check on that for me. This Expert Series will be recorded and placed on demand within 24 hours, so please let your colleagues know how you enjoyed the presentation today and direct them to the On Demand tab or sign up for future talks. So to get everyone a little familiar with the chat function, I want to see where everyone is tuning in from. I'm in New Jersey. Gavin and Melania, where are you guys at?
We are both located in Solon, Ohio, at our Solon facility from MP. I am currently traveling in China right now, but Melania should be in Solon facility right now.
Yeah, seeing she has Cleveland, Ohio in the chat. There's also Philly, Colorado, Georgia, North Carolina, and Puerto Rico in the chat hanging out with us today. So I'll go ahead and turn it over to Gavin and Melania here today to talk about MP Biomedical, food testing, and plant research solutions.
All right, can everyone see my screen?
Yep.
All right, perfect. All right, thank you everyone for attending the meeting. We really appreciate. My name is Gavin Wong. Me and my colleague Vanania today are going to talk about the food testing and the plant research solutions that we are offering at MP Biomedicals. So this is me and my colleague, and Melania will start with the SafTest portion, and later on I will be following up with our FastPrep system, which is the homogenizer that's assisting your plant and animal research in terms of sample preparations. Without further ado, I will hand it to Melania on the SafTest portion.
Okay, thank you, Gavin. So this webinar, as Gavin mentioned, will cover SafeTest, specifically food quality testing is where that falls. And it'll just I'll cover why it's important, the challenges, and what the SafeTest solution is. And then Gavin will talk about sample prep, food testing, and plant research. And he will talk about sample preparation, current challenges, and the FastPrep beating workflow solutions. Next page. Okay, so the importance of food quality testing. So when you talk about the importance of it, one thing that comes up is texture, which is like deterioration or the emulsion breakdown. And the best way I can describe that is when you leave a bag of chips open overnight and then you go to taste the chip and you can taste and tell that the chip is stale. Then you have your sensory, which involves rancidity, off flavor, odor, or maybe a darkening of the material. Then there's the lessening of the nutritional value. That's the loss of nutrients or essential components. Product loss of consistency, reliability, stability, and then the safety piece is the reduction in shelf life and the ability to actually consume whatever that food option is. Next screen, please. So the challenges when it comes to food quality testing. So if you wanted to do the AOCS method, that will require a large amount of sample. The usage of toxic chemicals such as chloroform. So if you're using that, you're looking at having an actual hood, ventilation, and storage for those chemicals. And it can take up to 18 hours to just extract the oil from the sample in order to even get to the testing portion. Another option is sending your samples to an outside lab. And the issues there is similar to with the AOCS method. You still need a large amount of sample. A lot of times when you're sending to an outside lab, they want enough to at least repeat the test 2 times. So you're looking double the sample amount. and it'll take 2 days at least to get your results, and then there's a high cost per sample. Next page, please. So we have our SafTest solution, which gives you the option to do the sample testing, and it's quick, it's easy, and you can determine the quality and freshness and raw materials and finished food products. Next screen. What is SafTest? It is a benchtop system used for analyzing fat and oil degradation in different sample types. It has the ability to test both liquid and solid samples. And after your initial capital expenditure for the platform and the other components that go with it, you're looking at about a $12 per sample, each sample that you test. Next. SafeTest capabilities. So SafeTest, we currently have 6 endpoints that we offer. The first 4 are in that degradation category. So that's peroxide, free fatty acid, malonaldehyde, and alkanol. We also have the ability to do percent fat testing, which is useful by itself, but also in a lot of different industries. Peroxide and free fatty acid, they want those reported on a fat basis. So you can use our percent fat kit in order to determine that. And we also have a test that can test the glycerin and biodiesel, and that would be our total glycerin kit. Next page. SafeTest application and samples. So you can use SafeTest to test the fat and lipids in oil, vegetable, olive oil, coconut oil, butter, milk, cream, seeds, grains, nuts, snacks and baked foods, fried foods, eggs, etc. We also are pretty big in the pet food space, so you can use it to test lipids in finished goods, foods, treats, raw materials, meat meals, fresh organs, feathers, pretty much anything you see in that rendering space, you can use it as well. And there's also lipids in consumer goods, such as cosmetics and skincare. Next screen. So the advantages of SafeTest. So you're looking at rapid on-site results. You can get your results in less than 20 minutes. There's no special lab or degree needed to run tests. There's no toxic chemicals, so you do not need a hood, special ventilation, or full lab setup in order to run this instrumentation. Each platform, they're flexible and customizable for the customer's needs. And there's an AOAC certification that our test kits hold. And that's just basically a validation that says that our results are comparable to the results that you would get using the standard AOCS method. Next. Okay, Gavin, I think this is you.
Sorry, I was on mute. I was saying that before we jump into the FastPrep portion of the presentations, do you want to do a really quick Q&A, or do you want to leave that at the end of the presentation?
I'm fine either way. I didn't know what the plan was in regards to that, so I would defer to.
Let's leave that to the end.
Okay.
Okay, perfect. All right. Thank you, Valenia. I will take over for the sample prep portion. So another product that MP offered is our FastPrep system. It is a homogenizer, which is used for your sample preparation process for your plant or animal research during your to homogenize your sample as a sample prep instrument for your workflow in terms of the research. So what is a sample preparation? when we were talking about the sample preparation in this presentation, we were talking about the biological sample by homogenizing tissues and analyzes the cell to extract your DNA, RNA, protein for purification and downstream analysis purposes. So for a lot of the research that we are doing right now, that is when you are trying to extract your DNA or RNA, you need to basically break the cell walls and release the DNAs and RNA out in order for your later on workflows. And in terms of the sample preparations, we are talking about the steps of homogenizing or using using a way to basically help you break the cell walls and release your nucleic acid out for your later on workflows. There are several ways on the market that people are using for sample preparations. In terms of the mechanical ways, basically using physical forces to break the cell wall. There's also pressures. There's also temperature treatment. There's also ultrasonications or the chemical way. So there is multiple ways of actually extracting your DNA and RNA out from your samples in terms of the sample preparation method. The FastPrep system is more a mechanical way of breaking the cell walls and release your DNA and RNAs. Later on, I will be sharing more information on how this is, how our system is a little bit better compared to the other ways on the market, which is in terms of temperature or ultrasonications or chemical ways of producing a better result with higher molecular weight and more reproducibility and have the ability to basically have a higher throughput comparing to the other ways. In terms of the challenges because in terms of our instrument as a homogenizer, we are consistently working on the market to see what is our we want to help our customers to overcome their challenges in terms of the sample preparations. And we have been summarizing them into a few very key points. We have encountered the problem. We often see our customers encounter the problem of either the sample quality, which you may have purity issues, and when you're using manual method, for example, like handheld homogenizer or a ultrasonicator, it is time consuming, there's inconsistency in the results, and there's less reproducibilities. There's also the risk when you are using a handheld system, which is a more open system. There's possibilities of contaminations during the homogenization process, and there's also other issues with so for example, when you are using a chemical ways to do the degradations, there's problem with the concentrations. You're unsure about how many what's the concentrations of the digestive buffers you should put into the when you are treating with different samples with different sample sizes, and there's basically inconsistency and unpredictable effect for your downstream applications and adaptabilities. So there is different types of samples. So for example, even with the tissue samples or the plant samples, you will be encountering hard samples versus the soft. Sometimes when you are doing the sample preparation, treating a tumor samples will be very different versus a just a normal liver sample, because the hardness of the tissues may affect your your sonication process, the time of your sonication process, or the concentrations of your digestive buffers to get to the results that you really want. And manual way wise, we also taking considerations of the throughput as in recent years, many scientists are using the have the need for more high throughput solutions with treating multiple samples at the same time and seeking for the reproducibilities. So these are all considerations and challenges that we gather from our customers in terms of the sample preparation step for their research. So in terms of our instrument, we are here to help to solve that particular problem. Our instrument is in particular designed as a bead beading using the bead beading technology to assist you overcome the challenges that I just mentioned in the sample preparation step. So this including, as I mentioned, this is a homogenizer. So it's the homogenized instrument, and it comes with the consumables of the tube contains the specified lysin matrix beads, which you will put your samples into the tube with the specified lysin matrix beads that is fit for your basically the hardness of your samples, and will be put into the instrument. The way it operates is that it's shaking the sample in an optimized motion, resulting in the collision of the sample material with the lysin matrix. So during the shaking process, the beads contains in the lysin matrix tube will homogenize basically your sample based on different sample type. There will be, different lysin matrix designed specifically for that. So some of the advantage, as I mentioned earlier, so versus a handheld homogenizer or a sonotator, it is much quicker, easier, and more convenient. And it is more, because as I mentioned, there's no further chemicals added. So there won't be any sort of like chemical needs or enzymes that could interface the downstream process for some of the samples. As I mentioned, it is a little hard to control for the concentrations versus your digestive buffer concentration versus your samples. In terms of ours, we are pure mechanical, so there won't be any effects of your digestive buffer effect to your sample type. It is also a closed system, so no cross contaminations. Each individual tube process one sample, so there won't be any cross contaminations across different samples. There's also flexibilities. It is also a high throughput. In terms of the throughput wise, we do offer the standard throughput is a 24 throughput, but you do have the options to upgrade it to 48 throughput with 48 2-ml tubes. We do also have the adapters. If you are processing higher volume samples, we do also offer 15 ml. 4.5 ml, 15 ml, and 50 ml options for you if you are seeking higher throughput options. So we covered, we also have a variety of lysin matrix, which covers different sample types, and the adapters will be covering your need for different volume of samples. In terms of the workflow wise, we do at MP, we not only offer just one bead beading system, so we do also offer a workflow wise solutions for your specific applications. So it is pretty simple steps. You will be basically having your samples and the FastPrep system, which you do have the options. As I mentioned, is 24 is the standard. But you can also upgrade it to 48 or up to 192 with our FastPrep 96 system. The system will be always combined with our lysin matrix tubes, which is the consumables that we'll be using on the instrument. And in terms of the purifications, we also offer nucleic acid purification kits and extraction kits. Or if you are using FastPrep 96 and you are seeking for a more automated workflows, we also offer our Mediflex series, which is a automated and nucleic acid purification system. And you will get once you're using the entire system, you will having better results that will be shown in the later slide for your downstream analysis for your PCR, qPCR, or the NGS workflows. So in terms of the product-wise, we do offer 4 options, which is your Super FastPrep 2. This is a low throughput field work using instrument. This is a handheld system, which you can just handheld it, and the throughput is 2. So each time you can process up to 2 samples. This is more for a field workers, which you because it doesn't need to be, it's just more designed in a way of the flexibility wise, which you can bring to the field. And if you want to process some samples, in the cornfield, you can print this, and it's not, it's designed for basically immediate need for some testing if you need to perform that in the field. In terms of the lab bench wise, we do offer FastPrep 24 Classic and 5G, which are the 2 top selling instruments in our product lines. So these 2, the FastPrep 24 is the 24 throughput one, and for FastPrep 24 5G, you have the options to upgrade to 48 2-ml tubes options, which you can homogenize up to 48 samples per time. So these are the 2 options that we offered in terms of the standard throughput, mid throughput, and in terms of the high throughput, we do have the options for FastPrep 96, which you are able to input the 2 96 default plate in there and perform homogenizations using that. So the FastPrep 96 can process up to 192 samples per time. This is our high throughput options. If you have the need for a lot of samples, the high throughput samples, you are able to use FastPrep 96. Our beta beating systems usually process sample very quick. So as I mentioned, you are able to input either 22, 24, 48, or 192 samples a time. And the usual process time is about ranging from 40 seconds to 120 seconds. Within this time range, most of the samples is able to complete the sample preparation process, and your samples is ready for the next step of usually we'll do a cryocentrifuge, and your DNA and RNAs will be released into the buffer that you input into the lysine matrix tube, and you can use that after your centrifuge. You can use that buffer for your downstream, extractions or other workflows that you want to perform. In terms of the lysine matrix selection side, our beads have a varies of the size, shape, and components. So it can be used for different settings as well as different sample sizes. So we do, as our homogenizer is being marketed over 2 decades, so we do offer a wide variety of different lysine matrices. And here is basically a chart for assisting you to select the lysine matrix that you want to use to process your samples. Here we want to share in terms of this chart is with more aggressive, and basically if you want to do more sharing or less aggressive sharing, or also depends on the hardness of your samples. We do offer different kind of the selections on what kind of lysine matrix you will be selecting. If you are ever interested, on our website, we do have a more complete guidance on which type of lysine matrix you will be selecting for your samples. There's a more in-depth sample lysine matrix selection guide over there to assist you to select the samples that you want. Here's just a quick another references on in terms of the selection wise. Usually for plants and tissues, we do recommend lysine matrix DNA, but also if you are also working specifically with other samples, we also have other lysine matrix designed for that. But this is a really quick chart to assist you to select the samples that you select the lysine matrix that you want. This information is also available on our website, so feel free to check that out. In terms of the sizes, as I mentioned earlier, we do offer the options for 2 ml tubes, 4.5 ml tubes, 15 ml tubes, 50 ml tubes, or the 96 depot plate for high throughput options. So these are all available, depends on your sample sizes, because we know for some research, your sample size is not limited to 2 ml tubes. So we do offer the options to upgrade to fit your workflows on amount of the sample that you want to process. In terms of the nucleic acid extraction kits, so these are additional options that we also offer to combine with our lysine matrix and bead beading systems. So this kit is specifically to utilize purified high yields of DNA and RNA from plants, environmental samples, and tissue samples. So we do offer animal kits as well as the plant kits. And it's being a long-standing kit on the market, and it's being combined with our bead beading system for a long time. Once you extract it from using our kits, it's ready to use nucleic acid for your downstream PCR to PCR, NGS, or other downstream workflows. There's multiple options for also for others. In terms of environmental samples, we do plant tissues, and we also we do offer a soil kit, which is also a top selling product within our company that also a lot of scientists use to extract it or isolate the bacteria from the food, saline wastewater, or other environmental samples. So this is specifically designed for if you are also interested in extracting DNA in specific, food samples or other contaminated samples. For if you are also considering a higher throughput, we do also offer a magnetic bead-based nucleic acid extraction system. This is an automated system that can process up to 96 samples, but this instrument also offers the flexibility of the single-stranded kits, which allows you to process 1 to 48 samples individually, or it's inputting the 96 depot plate, so you can also process 16, 32, 48, 64, or 96 samples at a time. This instrument also works individually, or it also works with our FastPrep system. So it is your options to combine. It doesn't, you don't need to be purchasing one in order for another one to work. It is so we do offer also the the extractions for the bacteria, fungus, processed food, plant, animal tissues. So these two instruments can be working together, or working individually, determined by if you need the need for a higher throughput, a combined more workflows, or just looking into individually a product that you want to either perform extractions, or if you are more interested in just for the simple prep set. So as a long-standing product, the FastPrep 5G itself do have more than 2,400 citations available. And for the FastPrep Classic, which is the smaller and older models, it also do have similar amount of citations on the market. So in combined, we do see more than 6,000 citations on the market currently. So there is most of the samples that you will be dealing daily, there is going to be a citation and specific workflows for that. For the time purposes, I'm just going to quickly go over a few. So this is the detecting pathogens in food and feed samples, and comparing the results of how the FastPrep system, in terms of once during the extractions, and the amount of the release from the whole cross section of the internal contaminated meat paste. As a beta beating system, compared to other methods, our system do provide you, it's more licensed completely, so it do provide you basically a better result when you are comparing to other competitors on the market, or versus the other method that I mentioned earlier, like the sonications, or using digestive buffer to digest your samples. Here is another examples case study of the evaluating performance of the FDA method for detecting the synthesis in fresh produce. This is also using our FastPrep system combined with Lysin Matrix E, which process usually another very popular to use to process plant and soil samples. As I mentioned, so this is a combination that with our FastDNA spin kit for soil to extract, and the results section showing the DNA was successfully isolated, and then showing the PCR detected detecting the specific things that you are interested within the samples. So in terms of the extraction side, again, as I mentioned, we do recommend to combine it with our kits in terms of the best performance. But our system is also designed to work with also if you're working with the trending kits on the market. So we do offer the flexibility over there. There's no like the saying where you have to use our systems with our kits. It will provide you with the best of performance. But if there's already a workflow set in the lab, for example, it needs to be using QIAGEN kits, the system does not affect the performance. If you're using other, the system performance not going to be affected if you're using other kits. So there is also others that due to time limitations, I won't discuss those in details, but feel free to check. Oftentimes we do recommend our customers, if you are really interested, it's pretty simple. All you need to do is log into the Scholars and type your samples and the FastPrep system in there. And 99% of the times, I guarantee you, there is going to be a publications which optimize the protocols already dead for that. As you know, we've been marketed for so long. There is a lot of R&D, a lot of scientists using our system and process that the samples that you are processing is the same sample you are processing today. So there will be a lot more citations. You can check it out. There is already pre-validated protocols. How many seconds, what's the speed of the instrument that you need to optimize for your specific samples. But if you did not find it, we're always welcome to reach back to MP Bio, and we are happy to assist you with the lysine matrix selections or the speed or the optimizations of the protocols that you want to use for your specific samples. But most of the times we do offer, there will be the citation already online, a pre-optimized from the scientists in your community, and it should be really easy, hands-on to use, as all the things are being pre-validated by a member in the community. All right, that is all the presentation we have today. Is there any questions?
So while people are taking the time to type out their questions, I'm going to go ahead and share my screen for just a moment.
Mm-hmm.
So I wanted to take a quick moment to show you that there is a giveaway if you take the survey inside of the kiosk. There's also literature, web links, as well as videos in here. So make sure you take some time after the talk and explore around. There's also the Thomas Center, so you can enter over here and check out some of these labs. There's also on-demand talks. So this talk will be on demand by tomorrow. So you can find that here as well as all of our past talks. So I'll go ahead and hand it back over to MP Bio to finish us out with some questions.
Okay, well, just wanted to jump in here real quick, and thank you everyone for joining. I'm Becky Burdick. I'm your channel sales manager here at MP Biomedicals, and I'm your single point of contact. So you can certainly reach out to the two presenters as well. We all work as a team. But if you need anything at all, you can reach out to me directly, and I'll go ahead and put my email address in the chat as well. So if you need anything in regards to pricing, literature, if you prefer not to click around, you just want me to send you something, or be happy to get on a call with your customer. And I did also want to mention that with Gavin's presentation with our FastPrep, we do offer demos, virtual demos, so we can actually bring your customer into a demo if they're interested in one of the FastPreps, and do a demo with your customer. We usually do a little kind of a question and answer with your customer first through email to kind of get an idea of what types of samples they use, what sample volume, and what their parameters are, so that we can tailor make the demo specific to their research as well. So just wanted to put that out there. And again, I really appreciate everyone's time and attention here, and look forward to working with all of you.
Thank you. All right, any questions? Oscar, I see your mouth moving, but you're on mute.
Oscar, we can't hear you.
Yeah, no questions. I just want to say thank you to Becky for all of her help with samples. I'm sorry that I was on mute, but I was saying thank you.
Oh, you're very welcome. Anytime. You're a pleasure to work with, and I really enjoyed actually meeting all the Thomas reps and Beth as well at the Summer Expo. So I'm really excited to be working with all of you, and it's a pleasure for me as well.
All right, thank you. And then I'll get back to you too, because I owe you some feedback. I'm still waiting for the feedback from my customer in Canada on those samples that you sent me. So I'll take care of that.
Okay, great. Looking forward to hearing.
All right. Thank you for a great presentation.
Thank you.
All right. Thank you all for joining us today, and hope you have a great rest of your day.
Thank you.
All right, thank you.